In this video, we're going to look at metabolic currencies. This is an idea that happens all across biochemistry, so let's just jump right into it. So there's a rule of thumb that running burns about 100 calories per mile. And that's a rough number, but it's good enough for the sort of thinking exercise we're about to do. If you ask, well, how much is 100 calories? Turns out that's about one slice of bread. It's a 0.4 Snicker bars. It's half of an IPA, and it's 7.8 charged iPhones. There's a fun party fact. Interesting question is, all right, if that's how much it takes to run a mile, how much ATP do we need to do so? And if you haven't heard of ATP at all before, ATP is a molecule that our cells use to do energy intensive things. So when you're contracting your muscle cell, you're taking molecules of ATP and you're converting it to ADP. In that process, releasing energy. In this case, you release 30.5 kilojoules per mole of energy every time you do that reaction. So that can help make reactions that are unfavorable thermodynamically favorable. So if you can get 30.5 kilojoules per mole out of an ATP, and ATP is 507 grams per mole, how much ATP does it take to run a mile? So this is one of those things, maybe it's worth actually pausing and thinking about and trying to do. I'm a big fan of participatory things, so why don't you try that? So go find your pause button and a pencil and paper and try to figure this out. So pause. Okay, we're coming back from our pause, and let's do this. It's a really simple, or I don't know about simple, but nice unit conversion problem. So it's 100 calories per mile. That's our number here. We can say that a calorie is 4.184 kilojoules. We know that it's 30.5 kilojoules per mole here, so we can say those kilojoules cancel. So now we have 100 moles of ATP per mile. We know that a mole of ATP is 507.18 grams, which gives us a total of 6,860 grams of ATP per mile. Cool. You're like, what is that? Well, this is about 7 kilograms. What is 7 kilograms? That's about 2 1-gallon jugs full of water. So 2 gallons of water worth of ATP. And if we want to put this into uncivilized American units, say it's 14-ish pounds. So a little more than 14 pounds of ATP to run a mile. We have a problem. And that problem is you have only about 100 grams of ATP. Numbers of what this is vary across the internet, but 100 is about right. It's within an order of magnitude. So how do you run, or how can anybody run a mile when they have something like 1.4% of all the ATP that they need, right? So they have 100 grams, they're going to use 6,860 grams. How do we reconcile these two things? The way we reconcile them is recognizing that ATP is not an energy source. ATP is a currency. It's a way to move around energy that we get from somewhere else, but it is not itself a source of any energy in cells. It's just a way to move energy around. Another way we can see this is if you just ask yourself, all right, if a typical diet or typical person burns about 2,000 calories in a day, that would mean they'd have to turn over every molecule of ATP by at least 500 times, probably more for many of them. But so there's every molecule of ATP gets charged and uncharged, charged and uncharged, charged and uncharged over and over and over and over throughout the day. It's not a source of energy. It is a currency. And so currencies are ways that cells move energy and other things around in small exchangeable units rather than as single big blocks of things. It's like a universal way to say, hey, I need a little bit of energy. Here's an ATP that has a little bit of energy. Now, when I was prepping this video, I sort of waffled back and forth. I was at one point going to do up to 6 different currencies, and I decided I would focus on just a couple in some detail, not deep detail, but focus on 2, and then we'll hit others as we go through future videos. And the currencies I wanted to cover were energy and electrons. So we've already seen the energy currency a little bit. This is ATP. ATP consists of an adenosine base, a ribose sugar, and then some number of phosphates attached to it. The ATP, adenosine triphosphate form, has 3. If we chop off one of these ATP molecules, that is a thermodynamically favorable step. We generate ADP and an inorganic phosphate. That is favorable by minus 30.5 kilojoules per mole. And there are a variety of reasons this is favorable. There's electrostatic repulsion between all these negative charges. There's also a difference in the resonance structures available. There's actually more resonance structures available in the cleaved form, so there's more ways to arrange the system. So just overall, it's more entropically favorable to chop off the phosphate. Okay, cool. What are the features of currencies? And it turns out they really all have three, and we can see where these how these are exemplified with ATP. So in the most technically, complicated, sciencey sounding vocabulary possible, these features are a business end, which I guess I'll talk about right now. You look at an ATP molecule, there's only one part of it that does interesting things, right? This side gets chopped off or not. The rest of the molecule doesn't change at all. So there's a small business end, a relatively small part of the molecule that does all the interesting chemistry. Second thing that they have is they have a handle for recognition. So if an enzyme needs to operate on ATP, it doesn't have to recognize just this little end over here. It's got all this other stuff to recognize. Like, oh, okay, that's an ATP. Cool, I can do something with it. So there's a handle to be recognized and to move the molecule around in the cell. Every currency has something like this. They've got a business end that's a small part, and then they have a handle that's a large part that the cell can use to recognize it. And the last thing about all of these currencies is that they are mobile. They can move around. They're not fixed in one location. This is a very water-soluble molecule that'll just float around. It can go anywhere in the cell, delivering bits of energy. Every currency we'll see across metabolism is going to have these features: a business end, a handle, and mobility. Now, another interesting feature about currencies that we're going to see, and we'll illustrate here with ATP, is that they work to keep the charged molecule about constant, and they measure the ratio of charged to uncharged species. So what do I mean by this? Well, the charged form of ATP is ATP. That's the molecule that's ready to go do interesting things. The uncharged form, there's actually two of them. There's ADP, so we've chopped off one phosphate. And there's actually AMP, which has only one phosphate. One phos, two phos, three phos. So what cells are going to do is they're going to try to keep ATP levels about constant, but they're going to measure the relative amount of energy they have as the ratio of ATP to these two molecules here. And so let's see this, for example. This is a biologically plausible. We have a cell that has an ATP to AMP ratio of 5 millimolar to 0.1 millimolar. This is a ratio of 50, so this is a high excess of ATP relative to AMP. If we take that cell and we put it under metabolic load, so now ATP is being consumed fairly rapidly, we'll actually see an interesting response. We'll see the ATP concentration doesn't drop very much. There's homeostatic mechanisms that work really hard to keep the ATP concentration relatively high, so we have a small change in our ATP concentration. But since cells are measuring the ratio of these two, we see a very large change in the ratio relative to that. So it goes from a 50-fold excess of ATP to AMP to now only a 7.5-fold excess. So a cell can say, oh my goodness, I need to make more ATP, even while the ATP concentration is remaining high. So cells work to keep the charged molecule concentration about constant. So they're going to keep ATP levels high, but they're going to measure how much ATP is around or how much energy is around as the ratio, which can change quite a bit. Okay, so that was ATP and AMP, also talking about some general features of currencies. Let's look at another currency, and that is our currency for electrons. So here's the main molecule that's involved in this, nicotinamide adenine dinucleotide, always said as NAD basically. This is, as I said, as the name says, it's a dinucleotide. We have an adenosine base over here with its phosphate, its sugar, and its base. And we have a modified base over here where we've got a ribose sugar, a phosphate, and then a modified base. And it turns out that this has the same features as an ATP molecule. There is a business end right here. This is where business happens. Basically a single position, and if you want to expand it out, we could say it's the whole ring, but there's really one place where we're going to see chemistry happening. All of the rest of this is a handle that can be recognized. And then if you look, this is a really water-soluble molecule that can happily wander around in the cell, so it is mobile. The way that nicotinamide adenine dinucleotide, NAD, works is that ring up here, the business end ring, can basically easily store electrons. So we can take 2 electrons and a proton, and we can stick it into this ring structure, converting from a positively charged aromatic ring into a non-aromatic neutral ring here. We've taken these 2 electrons, we've stored them in the system. In fact, we're going to be doing redox chemistry later. So we're going to be doing it later, but it's something we're going to encounter a lot. So let's actually do a little primer reminder on this and see what we mean by this is storing electrons. We can actually count pairs of electrons just by looking at the number of bonds. So here on the left, I've got 2 electrons. So I'm going to call that 1 pair of electrons. I've got 2 pairs of electrons in this ring. I have 3 pairs for one side of the double bond, 4 for the other. This is a double bond, right? So now I've got 4 pairs of electrons thus far. 5, 6, 7, 8, 9, 10. So I've got 10 pairs of electrons on the left. On the right, we can say, well, we've got 1 pair of electrons there, 2 pairs of electrons there, 3, 4, 5, 6, 7, 8, 9, 10. But we missed 1 pair, and that's the lone pair, 11. So this has 2 more electrons, 1 more pair of electrons on here on the right, as well as a proton. So we can use this. We can go over here. We've stored some electrons. Go back here. We pulled the electrons out. So this is a currency for electrons. Stores electrons. And just like ATP, what we'll see is this gets turned over all the time. It sticks electrons in, pulls electrons out. Sticks electrons in, pulls electrons out. Now, it turns out cells use NAD plus for oxidation, meaning that they like to have this molecule around, ready to take up electrons. And remember what we said for ATP, cells keep the charged concentration high. We see that there's an excess of the charged molecule. We have 100-fold ready to take electrons relative to already took up electrons. So it's a 100-to-1 ratio for that. Cells use a slightly modified molecule for reduction, and this is NADP. And hopefully what you can see there is there, I'm going to circle it and we're going to flip back a slide. The only difference between these two is that phosphate. And it turns out this phosphate is a handle itself. This lets an enzyme recognize, am I interacting with an NADP, which will have this phosphate on it, or am I interacting with an NAD? And that lets cells actually maintain 2 different sets of currencies, right? We have a currency for oxidation. That's what NAD plus is. This is giving you oxidizing power, the ability to go take electrons from things. This is used for reducing power. This is, I have electrons that I want to give away. And like we've said with keeping the ratio of charge, so the charged form of NADPH is the one with, or the charged form, the charged form of NADP is NADPH. So we see we've got a high amount of NADPH relative to NADP plus. And what this means is cells can hold on to this and use it for reduction. And once we get into redox chemistry, we'll really start unpacking that in a bunch of detail. But for now, hopefully what you're seeing is that we can have 2 pools. We can have NAD plus around at a high concentration. That's our currency for oxidizing stuff. And then we can have NADPH around also at a high concentration, and that's used for reducing stuff. That means we can have a pool for catabolism, for oxidizing things. And we can have another pool for anabolism, for reducing things. Okay, so hopefully what we've seen in this video is that this idea of currencies transfers, that we can think about ATPs, which you may have heard of described as a currency. We can apply it to think about things like electrons with oxidizing power, reducing power. We'll also see how we can apply this to different things like nitrogen levels or in some contexts, carbon levels. So it's a really nice universal feature of these systems. But just to quickly reiterate, what does a currency consist of? It has a business end, it has a handle, and it's mobile. And it's something that lets cells move around small units of something, energy, electrons, or something else, in order to do metabolism in an organized way. All right, so that's what I have on that. Hopefully it was helpful, and I will talk to you later.